Tip growth in plants by Brittany Tolliver and Lindsay Posley.
Tip growth is a type of cellular growth that is represented in many diverse cell types, which are important to plant breeding in the pollen tube, agriculture in the hair roots, and plant and animal pathology, such as in the fungal hyphae.
Tip growth is important because it allows different species of plants to occupy different niches. By occupying different niches, there's less competition, which allows many species to coexist at once.
Plants undergo exponential growth as long as the necessary resources are available. These resources include water, sunlight, carbon dioxide, and key soil nutrients. Here we have a model representing tip growth in a plant over 3 different growing seasons. The green is the first growing season, the orange is the second, and the red is the third. As you can see, over each season, the plant is continuously undergoing tip growth in the stalk, the leaves, and the root hairs.
Plants undergo a cellular level growth called elongation. Elongation includes 3 major parts: the cell itself, the microfibrils that are parallel to each other and wrapped around the cell, and the hemicellulose, which prevents the microfibrils from separating.
The plant releases a hormone called auxin, which stimulates the release of protons to create an acidic environment so that a protein, called expansin, can break the hemicellulose bonds. Oh, we lost it. Keep it rolling though. We're just going to take it.
Microfibrils then separate, which allows for the elongation of the cell. As this is happening, the plant is making new cellulose. Bundles of cellulose are what make up the microfibrils that surround the cell. 6 cellulose synthesizing enzymes are arranged together in a subassembly. 6 of these subassemblies are then arranged together in a larger structure called a rosette.
Each cellulose synthesizing enzymes makes 1 strand of cellulose. So each subassembly makes 6 strands of cellulose, and each rosette makes 36 strands. This positioning is what forms a microfibril. The rosette moves around the cell, making microfibrils.
Okay. These microfibrils must be kept parallel to each other, and there are 2 different theories for how this happens: the bumper rail model and the monorail model. In the bumper rail model, the rosette is pushed forward by the growing microfibril and is restricted to the lane between the adjacent tubules. The monorail theory is that the rosette is pulled forward by the microtubule motor proteins.
This concludes our presentation on tip growth in plants. Thank you.